Hey, this is the video for Molecules.3. So go ahead and set up your Cornell notes. You can open up to a new page and make your entry. Pay attention to the essential vocabulary. We're going to be talking about indicators, Benedict's solution, and Lugol's iodine. So let's begin. An indicator is, technically, it's just a chemical that changes color in the presence of a specific molecule. So go ahead and write this down in your notes. You will need this definition in the future. We're going to be discussing 3 different types of indicators that you might use in a living environment class. The first one is glucose indicator, and it's technically called Benedict's solution. So sometimes they might say glucose indicator, sometimes you might see the word Benedict's. If you remember that Benedict's is glucose indicator, you'll be fine. So we can use it to test different types of macromolecules. We can use it to test, there might be glucose in sweets, anything that is sugary. This represents meats, which is proteins, and this represents starchy foods, anything that would have carbohydrates. So if we add the glucose indicator to all 3 of these samples, it's not going to change color because you have to do something first. Glucose indicator is a little bit different because it must be heated. So you have to heat the sample after you apply the glucose indicator. Now when you heat the sample that was tested with fruits contains glucose, so it's going to be positive. And it will turn this like brick red color, like kind of like the color of, you could say orange, you could say a reddish, a dark red color. But basically the Regents likes to use the term brick red. Now if you put the glucose indicator in protein and turn on the heat, it's going to give you a negative result. It's not going to change. Same thing applies if you put it with starches or carbohydrates. It will give you a negative result and the chemical will remain unchanged. Now another type of indicator is a protein indicator, and this is also known as a biuret solution. So protein indicators are not used as often as the other 2, but you might see it. The protein indicator, so if we added it to all 3 of these foods, it has a kind of a deep blue color. When you add it to glucose or anything sweet with sugars, it's going to give you a negative result. There will be no change. When you add the biuret, the protein indicator, to meats, which contain protein, it will turn a purple color, and that indicates positive. And when you add it to starchy foods or foods with carbohydrates, you will get a negative result. There will be no change. And finally, the last indicator is starch indicator. And this is called Lugol's iodine solution. So iodine, you've probably used it if you ever had a cut. It acts as a disinfectant. But it also does something quite interesting when it encounters starch. So when I add the iodine or the starch indicator, it's going to have this amber color. And amber is kind of this, like, honestly, it looks like urine. So if you said it's pea colored, I mean, I would probably try to remember the word amber. But if you said it's yellow like urine, yellow like pee, I'm sure you'd get credit for it. Now, when you add it to foods that have glucose, it will be a negative result. There will be no change. When you add it to foods that have protein, again, it will be a negative result. You will see no change. But when you add starch indicator to foods containing starch or any sort of carbohydrates, you will get a positive result, and it will be a dark purple, or also the Regents calls it blue-black. So it will be a deep dark purple, almost looking black color. So again, this indicates a positive result. So I just gave you a bunch of information on indicators, and I hope that this was informative. Please respond to the check for understanding so we can ensure that you understood the materials that were presented to you. I hope that this video was informative. Thank you for your attention. I will see you soon.